Hello, my name is Shawna Hottinger. I work with the research at the Division of Allergy and Immunology as a medical writer and editor, so I see a lot of great science come across my desk. Here at Cincinnati Children's, we believe in sharing our ideas, our discoveries, and our advancements. Today, we're going to have a talk along about recent basic and translational research highlights from our division related to eosinophilic diseases. Mast cells and eosinophils are immune cells that are commonly found in human tissue biopsies, but their clinical significance during health and disease continues to be investigated. Our physician researchers were part of a work group that developed a standardized consensus for assessing the number and quality of mast cells and eosinophils in pathologic specimens taken from the skin, gastrointestinal tract, and bone marrow to benefit clinical practice in patients. This research was partially supported by the Consortium of Eosinophilic Gastrointestinal Disease Researchers, which is called CIGR for short. Expression of the transcription factor ALOS increases during the commitment of progenitor cells to the eosinophil lineage and during eosinophil maturation. Our researchers found that homing of circulating eosinophils to the tissue during healthy inflammatory allergic states depends upon ALOS, identifying ALOS as a potential therapeutic target for eosinophilic disease. Our researchers identified rare eosinophilic esophagitis-associated gene variants of desmoplakin, abbreviated here as DSP, and periplakin, abbreviated here as PPL. These variants impair the epithelial barrier. DSP and PPL are part of junctions linking cells together called intercellular junctions. Dysregulation of intercellular junctions suggests a mechanism whereby inflamed and hyperproliferative esophageal epithelial tissue can be penetrated by allergens, thereby amplifying allergic inflammation. DSP and PPL loss also occurred in non-familial eosinophilic esophagitis, suggesting that therapeutics to normalize intercellular junctions, and thus the epithelial barrier function, may reduce harmful outcomes of multiple forms of eosinophilic esophagitis. Interleukin-33 has a central role in initiating type 2 immune responses and is generally thought to be released from the cell following cellular damage and then processed extracellularly, meaning outside of the cell. Importantly, type 2 immune responses are known to be involved in eosinophilic diseases. Riboptosomes are composed of many proteins and act as signaling platforms to determine whether a cell will undergo cell death and by which process. Our researchers describe a novel riboptosome pathway that is assembled following exposure to various unrelated environmental allergens. This novel riboptosome pathway processes interleukin-33 into an active form intracellularly, so inside of the cell. This newly identified process is an epithelial allergen sensor, now called RIP-interleukin-33, and explains how environmental allergen exposures can lead to cell death and cell damage. Colonic eosinophilia is an enigmatic finding and poorly understood condition that is often referred to as eosinophilic colitis, or EOC for short. Whether eosinophilic colitis is a distinct disease or a colonic manifestation of eosinophilic gastrointestinal diseases, called EGID for short, or inflammatory bowel disease, was previously undetermined. In this CIGR-supported study, our researchers established eosinophilic colitis transcriptomic profiles, identified mechanistic pathways, and integrated findings with parallel inflammatory bowel disease and eosinophilic gastrointestinal disease data. These findings establish eosinophilic colitis as a distinct disease compared with other eosinophilic gastrointestinal diseases and inflammatory bowel disease, thereby providing a basis for improving diagnosis and treatment. Recent public media has focused on the importance of gut microbes in health and disease. Our researchers investigated fundamental properties of host microbiota interactions in the esophagus and established the composition, transplantation potential, antibiotic responsiveness, and host microbiota interactions of the esophagus. Fecal matter transplantation reconstituted the esophageal microbiome, opening up fecal transplants as a consideration for treating esophageal disease. antibiotic usage predisposed to allergic disease in an eosinophilic esophagitis experimental model in mice. These findings have implications for understanding health and disease in the esophagus and managing eosinophilic esophagitis. Mast cells are cells that produce multiple effects and accumulate in the esophagus of patients with eosinophilic esophagitis, called EOE for short. Mast cells are thought to contribute to disease pathogenesis, yet their properties and functions in the esophagus were largely unknown. Our researchers performed a comprehensive molecular and spatial characterization of esophageal mast cells in eosinophilic esophagitis and found that esophageal mast cells consist of diverse populations. These diverse esophageal mast cell populations had transcriptional signatures that associated with distinct spatial compartments and eosinophilic esophagitis disease status. The researchers found that mast cells assume a pro-inflammatory state and locally proliferate in active eosinophilic esophagitis, and importantly, remain activated and poised to reinitiate inflammation during disease remission. Eosinophilic gastrointestinal diseases are rare allergic conditions of the gastrointestinal tract, with eosinophilic esophagitis being the most common and studied. Importantly, diagnosis of an eosinophilic gastrointestinal disease is often delayed. We engaged in the U.S. Food and Drug Administration workshop hosted on July 21, 2021 to discuss the disease characteristics, natural history, and endpoints to assess treatment benefit in patients with eosinophilic gastrointestinal diseases beyond eosinophilic esophagitis, such as eosinophilic gastritis and eosinophilic colitis. Me and our colleagues published an article that summarizes and comments about this FDA workshop. For the FDA Grade 6 workshop on Aegis Beyond EOE meeting recording, please visit the National Institutes of Health's webpage for the event. The URL is provided on the screen. We believe that science and compassion are equally important. The Cincinnati Center for Eosinophilic Disorders leads the field with care and research of eosinophilic conditions in children and adults, and is the coordinating center for SEGR, part of the Rare Diseases Clinical Research Network of the National Institutes of Health. The Rothenberg CURED Lab, kindly supported by the Campaign Urging Research for Eosinophilic Disorders, the Sunshine Foundation, the National Institutes of Health, and Cincinnati Children, conducts basic and translational research of allergic and eosinophilic conditions. We believe that small acts have tremendous impact. Thank you for your interest, generous research support, and inspiring patient and family participation that have made this research possible.